everybody who is here attending, indeed, instead of enjoying the sun outside. I will do my best to keep this interesting for you. But indeed, my name is Sofia Prekatsounaki. I'm a PhD student at the Institute for Family and Sexuality Studies. And today, I want to tell you a story, the typical boy meets girl story, which of course goes something like this. Boy meets girl, or man meets woman. They fall in love. He thinks about sex every 7 seconds and is ready to get out at it anytime. She needs flowers and chocolates and candlelit dinners to get convinced. And of course, he does all that. She gives in. They cannot keep their hands off each other. They're dying to have sex again every chance they get. So this dying to have sex with each other, this exact subjective experience of an inclination to engage in sexual activity specifically with one's partner is what we call dyadic sexual desire. And we call it dyadic to distinguish it from solo sexual desire, which is the desire to be sexual with ourselves, and desire for a random attractive stranger. And dyadic sexual desire is what I will be talking to you about today. So the partners in our story, they feel sexual desire for each other. They are satisfied with their relationship, their sex life, and themselves. So they get married. They buy a house with a garden. They have 1.6 children and live happily ever after. And barely think about sex anymore. They miss the sexual tension and the sexual desire they felt for each other at the beginning of their relationship. But there's just nothing to do about it because their sexual desire just came falling from the sky back then, and it just doesn't anymore. So this is an old story. Unfortunately, it's still the typical story, while so much has changed. For one, the typical progression of a relationship, and certainly the gender composition of couples, but also what we know about the sexual motivation of men and women. And still, two things about this story remain timely and realistic. The first one is the decline of sexual desire between partners in a long-term relationship. And the second one is how partners feel that is out of their control. Regarding the first one, the decline of sexual desire, it is indeed very much in line with reality, because a decline in sexual desire is actually one of the most important, sorry, I don't know if somebody took over control of my presentation, but I'd like to do that myself, thank you. So the decline of sexual desire is the most often reported reason for seeking couple and sex therapy. And that is not so surprising. As Marta Meana, a prominent sex researcher, has put it, without the construct of desire, the sexual response seems incomplete and automatic. It lacks agency, and it does not align well with the observed and self-reported complexity of the human sexual experience. So that's why we try to get it back when it wanes, when it declines. And I would like to make a distinction here between decline in sexual desire and a decline in the frequency of sexual behavior. Because sometimes, indeed, couples seeking sex therapy want more sex with each other, but most often they want better sex, which is sex accompanied by sexual desire. And more sex does not necessarily imply better sex. Experimental research has in fact shown that simply increasing the sexual frequency in a couple does not increase sexual or relationship satisfaction, sometimes even quite the opposite. On the contrary, longing for sex, desiring sex with our partner, does relate strongly to our well-being, personal, relational, and sexual. However, as mentioned, sustaining it in a long-term relationship seems to be a challenge. So this challenge was the first aspect of our story that remains timely, realistic, and relevant. The second relevant aspect of the story is related to the feeling that sexual desire is beyond our control. And this uncontrollable character makes it mysterious. Until now, sexual desire has indeed been considered an enigmatic urge. Just as hunger or thirst, it was supposed to arise from an instinct, a primal need that just demands to be satisfied. but is totally out of our conscious control. In qualitative research that we have conducted, our participants, men and women in committed relationships, did indeed mention this uncontrollable, mysterious part of sexual desire. And it seems to be a part that we hate and love at the same time, because it makes the experience of sexuality special. It adds magic to our connection, to our sexual connection with our partner. But the participants in our study I was just talking about did not only talk about the mysterious part of sexual desire. In fact, they mostly talked about tangible and identifiable prerequisites and triggers for sexual desire, and even about strategies they deliberately use in order to manage their sexual desire for their partner. So the stories were mostly stories about agency and control over sexual desire. And when it comes to these tangible, identifiable prerequisites, triggers, and strategies for sexual desire, some of them were intrapersonal, relating more to individual processes within the individual. But more often they were interpersonal, relating to interactions between two partners. This resonates with clinical approaches of sexual desire problems that are increasingly in the clinical context are increasingly approached as a dyadic problem. So dyadic partner interactions are increasingly the focus of clinical approach of these problems and sex therapy. Also, accumulating empirical evidence highlight the role of such interactions for sexual desire. So, for example, we know that intimacy or perceived sexual compatibility are indeed factors that have an effect on our sexual desire for our partner. Unfortunately, these clinical and empirical insights are not incorporated yet in theoretical models. They are still fragmented, and thus they limit our understanding of how sexual desire really works in a dyadic context, which is the context of two partners in a romantic relationship. So, in order to advance this understanding of the dyadic nature of sexual desire, we propose a new framework, a new model, which we suitably call dyadics, and which stands for dyadic interactions affecting dyadic sexual desire. We propose that these interactions, which are relevant for sexual desire between partners, take place in 3 domains, each of which entails interactions focusing on one basic human need. And these domains are the domain of sexuality, of exploration, and of affiliation. Interactions in the first domain, the domain of sexuality, are focused on sexual needs. They relate to sexual attraction, sexual gratification, and sexual pleasure. Interactions in the second domain, that of exploration, are focused on the basic human need to explore and grow. They are the least studied, actually, in relationship to sexual desire, but they relate to cognitive curiosity, our innate motivation to seek new information about oneself and the world, and the motivation to learn and grow. Last but not least, the interactions in the third domain, the domain of affiliation, are focused on the need for connection. And they are probably the most often studied in relationship to sexual desire, and they relate to companionship, intimacy, mutual support, and also exchanges of affection. Now, in trying to satisfy these needs, we shape, according to dyadics, our interactions with our partner in 3 ways: by taking, giving, and receiving. Taking is about attending to our own needs, making sure that we fulfill them. Giving is about attending to our partner's needs, the responsiveness to them in the different domains that we just discussed. And of course, as everything in life and relationships, it seems important to find the right balance between this giving and taking, between this focus on our own needs and our partner's needs. Empirical findings suggest, namely, and this is a spoiler alert, that they both have a beneficial effect on sexual desire. But when we focus on our partner's needs so much that we sacrifice our own, the effect on our sexual desire is not so positive anymore. So balance between taking and giving is important. But we also have receiving, and receiving reflects what we feel we get out of our interactions with our partners. Based on theory and research, there are two things that are important to receive. That is responsiveness to our needs and validation for our sense of self in each of these domains, the sexuality, exploration, and affiliation. So to conclude, in our interactions, we take, we give, and receive. And of course, so does our partner. And Dyadic's main message is that what each partner takes, gives, and receives in the domains of sexuality, exploration, and affiliation is what determines their sexual desire for their partner. But what does that look like? Taking, giving, and receiving in these domains. And of course, what do we already know about their effects on the sexual desire between partners? Well, we start with the domain of sexuality. When it comes to taking in the domain of sexuality, a relevant construct is that of sexual subjectivity. And sexual subjectivity is all about understanding oneself as a sexual being. It is about feeling desirable as a sexual partner, but also feeling entitled to sexual pleasure and also being able to do what is necessary to experience sexual pleasure. For example, being assertive in our sexual interactions with our partner. It may not come as a surprise, but sexual subjectivity has been repeatedly found to correlate positively with sexual desire, which means that when we make room for our sexuality, for our own sexual needs, we also enable ourselves to feel sexual desire for our partner. But of course, it's not only about our needs. Research on sexual communal strength, which is the willingness to meet our partner's sexual needs, has also revealed positive ties with sexual desire. That means that being open to the sexual needs of our partner, be willing to meet them and satisfy them, also creates a bridge for us to feel sexual desire for them. In fact, the willingness to meet our partner's sexual needs has been identified as a protective factor against the decline of sexual desire in long-term relationships. And last but not least, what also is important for our sexual desire for a partner is how they react to our sexual needs, or actually how we perceive them too, what we feel we receive from our partner in our sexual interactions. As explained earlier, this receiving has to do with responsiveness and validation. So perceived responsiveness of our partner in the sexual domain is about feeling that our partner responds adequately to our sexual needs. Empirical evidence is still scarce, but theory does suggest that having a partner that satisfies our sexual needs will have a positive effect on our sexual desire for them, because we feel that having sex with them will be a positive or a rewarding experience, so our desire to do that will be strong. So receiving responsiveness seems to be a good thing. But receiving validation is also very, at least as good. And by validation in this domain, the domain of sexuality, I mean the acknowledgement, the confirmation of our sexual worth and our sexual self by our partner. In other words, the feeling that we are desired by our partner. So to summarize, the Dyadic model suggests that when we take the time and space necessary for our own sexual needs, when we give attention to our partner's sexual needs, and when we receive responsiveness to our sexual needs from our partner, we will feel strong sexual desire for them. Then we move to the domain of exploration, because besides our sexual needs, it seems to be important to also pay attention to our cognitive curiosity and growth needs. And that's exactly what the second domain is about. So taking in this domain refers to asserting one's own exploration and growth needs in order to reach a sense of self-efficacy. Clinicians and empirical researchers acknowledge that being able to pursue such autonomous growth is beneficial for the sexual desire we feel for our partner in a relationship. What also seems to be beneficial is when we get the chance to fulfill these needs through our partner. For example, by learning something new from them, or taking advantage, in a good sense, of their unique capabilities in order to expand our potential efficacy. But just as with interactions in the domain of sexuality, taking is not the only relevant contribution to our partner's interaction, but giving is also important. So giving in this domain of exploration has also been linked to sexual desire between partners. And what research suggests is that making room for the growth needs of our partner, either by creating opportunities for couple growth, so to grow together as a couple, or by facilitating our partner to autonomously pursue their own growth goals, serves our sexual desire for them. And last but not least, receiving the same responsiveness for them, receiving their support for our personal projects, perceiving their affirmation and appreciation for us as cognitively curious beings seem to be a powerful aphrodisiac in a romantic relationship. So to summarize, what Dyadic proposes regarding partner interactions in the domain of exploration is that our sexual desire for our romantic partner will be the strongest when we pursued autonomous and couple growth, and when we and our partner, facilitate each other's pursuit of exploration and growth needs. And now we move to partner interactions in the domain of affiliation, the last one. And of course, taking, giving, and receiving is again important here. When it comes to taking in this domain, we refer to one's needs and active pursuit of intimacy and closeness with a partner. Research has repeatedly shown that individuals who seek and facilitate intimacy with their partner also feel strong sexual desire for them. But how may we do this as partners, seek intimacy? Well, one of the ways that is research studied in relation to sexual desire is self-disclosure. That means sharing very personal information with our partner, letting them in our emotional experience. And that has a positive effect on our sexual desire for them. But just as in the domain of sexuality and exploration, also here it seems to be beneficial to focus not only on our own needs, but also on the needs of our partner, their emotional needs in this case. So feeling protective of a partner, for example, or expressing affection towards them has been found repeatedly to correlate positively with sexual desire for them. And not surprisingly, next to giving affection, receiving it also has an effect on our sexual desire for our partner. So when our partner is again responsive to our emotional needs, for example, when they support us in a difficult situation, when they listen to us and try to understand our worries or share our enthusiasm, when they express their love and appreciation for us, that is, according to science, when we are most likely to feel sexual desire for them. Thus, in the domain of affiliation, evidence shows that our sexual desire for our partner will be the strongest when we seek connection with them and when we give and receive affection and love. It sounds pretty simple, right? And maybe a little bit too simple. If that's your impression, you are right. Because while this closeness, affection, and intimacy between partners has unquestionable ties with relationship satisfaction, there is clinical and empirical evidence that it can be detrimental for sexual desire between partners. And also among laypersons, I think everybody has heard before proverbs like fire needs air, or ideas like you only appreciate something when you lose it. So the question is, when we are very close with our partner, can we desire what we already have? That's the question. And according to Dyadics, it is possible. We can desire what we already have. And that is possible when partners find the right balance. And that balance is between taking, giving, and receiving, between the focus on our own and our partner's needs and what we receive from our partner, but also balance in our needs in the different domains we identified here. So the domain of sexuality, exploration, and affiliation. It is when there is a balance between autonomy and connection, and a balance between experiencing ourselves and our partners as sexual, but also as emotional and cognitively curious beings, that our sexual desire for each other will flourish, according to Dyadics. And what about the long run? Although Dyadics has not been studied longitudinally yet, it does, of course, focus on dynamic interactions between partners. And that's why it is well suited to explain the ebbs and flows of sexual desire within a romantic relationship. And as mentioned in the beginning, empirical evidence shows that sexual desire declines in long-term relationships. And such decline has often been explained based on theories of relationship development. And they suggest a typical transition from passionate to companionate love, with passion and sexual desire typically prominent in the first type, at the beginning of a relationship, but not in the second. So, for example, the honeymoon phase, as it is often called, the first phase of couple development, is said to be accompanied by strong feelings of sexual desire between partners, while later relationship stages they are expected to have more sexual problems. However, research also suggests that sexual desire is sustained or wanes depending on how couples cope with transitional challenges that come with duration of their relationship, and that relational interactions may prevent sexual desire declining for one's partner. Also, studies finding that relationship duration is not a significant predictor of sexual boredom, and reports of increasing trajectories of dyadic sexual desire point to a variance in the often observed trend of declining sexual desire. So empirical research adds important nuances in this. And it suggests that the typical attenuation of dyadic sexual desire has something to do with the duration of the relationship, but it is more possible that relationship length as a risk factor, it's more about the factors that accompany relationship length than the relationship length itself. So according to Dyadics, important factors that may accompany relationship length and affect dyadic sexual desire are related to interactions between partners. The idea that relationship length brings about changes in these interactions is supported by theoretical and empirical evidence. Like, for example, we know that this honeymoon phase I just mentioned is accompanied by mutual support, positive loving behaviors, verbal and non-verbal exploration of sexuality, etc. While later relationship stages are accompanied by increased responsibilities, reduced couple time, physical health conditions, etc., etc. So the dyadic interactions that typically accompany relationship initiation are interactions that have been found to facilitate dyadic sexual desire and decline as relationship progress. On the other hand, the interactions that accompany relationship length are interactions that have been found to dampen sexual desire. So based on this, we propose that the dampening of dyadic sexual desire that comes with relationship duration, if it comes, may be attributed to a changing relational context and changing interactions between partners. So it is not relationship duration itself, but the changes in those partner interactions that are significant for dyadic sexual desire, regardless of the stage of their relationship that they may manifest themselves. That means that regardless the stage of their duration of their relationship, when dyadic interactions in the domain of sexuality, affiliation, and exploration become dysfunctional, the dyadics model predicts that dyadic sexual desire will become dysfunctional too, and the relational and partner interactional prerequisites for its development will wane, so sexual desire itself will wane too. Which brings us to the most important message and the takeaway from this talk, which is a constructive and more hopeful view of sexual desire in a romantic relationship. This view is that sexual desire is not an enigmatic urge that comes falling from the sky in the beginning of a relationship and then just disappears in the long run. Neither its activation nor its decline is outside of our control. On the contrary, according to Dyadics, sexual desire in a romantic relationship is tied to tangible and dynamic interactions between partners, interactions that are within our locus of control. And that, we believe, creates a world of possibilities for sexual desire, and with that, for sexual and relational well-being to be found and sustained in the context of a long-term romantic relationship. Thank you very much for listening.
Thank you very much, Sofia. I think a great presentation with a limited number of words on your slides. So you were really forcing us to listen to your positive story during springtime. So sometimes they say love is in the air, but I think it's good to know that there's hope. Are there questions from the floor? Maybe some quick questions before we move on with the next speaker. So you were saying that there are tangible aspects. So you presented a very nice overview that there are a lot of areas where improvements can be made. Is there like one aspect or one or two things that stand out where success is the most to be expected, or is it just the whole picture that counts?
Yeah. Yes, thank you very much for this question, because it's interesting to say, well, actually, what we already have found from our own research on this model, it's not finished yet. We are currently testing it, but the first evidence shows that all domains of partner interactions and all taking, giving, and receiving therein are all very important as prerequisites for sexual desire. But when you look at the relative predictive value of each of these interactions, it seems that interactions on the domain of sexuality actually have a stronger predictive value for sexual desire. So although it is very, very important and it is a prerequisite for partners to have satisfactory interactions in the other two domains, it seems that sex is about sex. The sexual desire is about sex. So the other domains really need to be addressed and to be included when we try to treat sexual desire problems. But it is also necessary to keep looking at sexual interactions.
Yeah. Okay, thank you so much. So more to come once your results are there. And are you planning to do a path analysis just to distinguish between the most prominent aspects and the other aspects?
Yes, among others, indeed. We are trying through qualitative and quantitative research to understand a little bit better how these domains interact with each other and how they affect dyadic sexual desire.
Okay. Thank you so much, Sofia. And I'm sure if there are other questions from the audience that you're happy to respond via email or a coffee in a nice sun. Okay.
Yes.
Thank you so much.
You. Thanks for having me.
I also see that our next speaker, Shen Zhong, is online. Shen, I already made you a presenter, so I think you can try to share your slides.
Okay.
Would you mind if I make a, yes, we can see it. Would you mind if I make a recording so that people can listen to it again, or people that cannot participate? So is that a problem to you, or you prefer not to have it recorded?
It's okay.
It's okay. Okay. So let's start on with your presentation. I don't know if you can switch your camera on, if that is possible. A completely different angle on the use of microCT tracking of human periodontal ligament stem cells labeled with gold nanocomplexes. I must admit there are a lot of difficult words to me, but I'm sure you will convince us about the social impact and the relevance of your research topic. Floor is yours, Chen.
Okay. Yeah. Okay. Yeah. Dear professors and colleagues, thank you for attending my oral presentation. I'm Chen Zong, a fourth-year PhD student in orthodontics, Department of Oral Health Sciences. I'm very interested in orthodontic tooth movement, bone regeneration, and stem cell research. Today, I'm very honored to stand here giving my first departmental lecture to you. The topic is in vivo microCT tracking of human periodontal ligament stem cells, labeled with gold nanocomplexes. Proud to say, this study has been accepted for publication by the Journal of Advanced Healthcare Materials with an impact factor of 9.93. Sorry. Yeah, generally speaking, the study design is actually quite perlusive and straightforward, as shown in the table of contents. Our research presents a breakthrough to label the periodontal ligament stem cells, which is also called PDLC. You just need to know it's a kind of dental stem cells. We can label the cells with optimized gold nanocomplexes, which can render the cells radiopaque and distinguish them from the surrounding tissue by CT, ex vivo and in vivo. The ex vivo means experimental outside of a living body, which is normally in the lab, and in vivo means experiment inside a living animal body. Briefly, in this study, first we synthesize the gold nanocomplexes, isolate human periodontal ligament stem cells in the meanwhile, and label the cells with the freshly made gold nanocomplexes. Then we checked whether the labeling can influence the viability and the differentiation capacity of the host cells. In the end, we tested the ex vivo and in vivo cell tracking by the gold nanocomplexes. Okay, now let me introduce the background. Because the PowerPoint I upload, there is no animation, so there is some superimposition of the pictures. Sorry for that. As orthodontists, our goal is always to shorten the time and reduce the side effects of the orthodontic treatment. As shorter orthodontic treatment time and less alveolar bone or root resorption are of great interest to both clinicians and patients. And that's why it's so important to investigate the biologic basis of orthodontic tooth movement, although it's quite like basic science. Recent research suggests that orthodontic tooth movement is not only an inflammatory reaction, but also includes the recruitment of mesenchymal stem cells in rats. And second, the periodontal ligament, PDL, can orchestrate the tooth movement by bone formation and resorption, because the tooth movement is actually a result of bone formation and resorption, the bone regeneration. And the interesting thing is there are a lot of mesenchymal stem cells in periodontal ligament called PDLSC. If we take this information together, it will be very interesting to investigate what the role of PDLSC in OTM is. As we know, PDL-SC are widely involved in dental and maxillofacial physiologic and pathologic process, not only in orthodontic tooth movement, but also in apical root resorption, tooth autotransplantation, alveolar bone loss, regeneration, and so on. And this process happened in many of dental diseases like periodontitis and implantitis. So for now, no matter if you want to investigate the role and the behavior of PDL-SC in these diseases or explore their potential for stem cell therapy, it is crucial to label the cells in order to monitor and track them in vivo. So developing a reliable and efficient labeling technique to track those cells is very important in basic science or in the prospective clinical study or application. So this labeling technique might be widely applied in oromaxillofacial research. Currently, there have been some labeling materials such as the GFP, BRDU, and spins, which can be used to label such cells in vivo. However, the GFP and the BRDU are dependent on immunohistochemical analysis, which means we have to euthanize the animals after the study to see something happen inside their tissue. This limits their application in repeated measure studies, because if we want to track how those cells behave in vivo, we cannot do it with those labeling agents because it needs us to kill the animals in the end. The last labeling agents, the spins, are trackable only in magnetic resonance imaging and have potential cytotoxicity, which also restrict their long-term application. So such agents are therefore not ideal for longitudinal in vivo tracking of our PDL-SCs. Luckily, based on recent literature, the gold nanoparticles is a novel nanomaterials which can be used to make the mesenchymal stem cells visible by micro-CT. This is very useful because micro-CT are widely used in dental and maxillofacial research to evaluate the hard tissue. And now we can also use this machine to track the labeled cells. It's a great advantage to avoid doing immunohistochemical analysis, as I mentioned before, which means if we do animal experiment, we don't need to euthanize the rats at different time points anymore. In such a way, we can reduce the sample size of the animal studies, which is good for us and also for the experiment animal. And also it can allow for longitudinal follow-up within the same animal at different time points. In the same way, if in the future we want to do the stem cell therapy on human, this labeling technique might also help us, because if we want to track how the stem cells influence our treatment on human, we have to take CT at different time points. If we have to use the GFP or BRDU, which needs immunohistochemistry analysis, we have to collect the samples from the human, which is not very human. So if we can use the gold nanocomplexes, it can allow for longitudinal follow-up, which means we don't need to collect anything. We just need to take the CT, and we will know what is happening in the human. However, this study has a drawback. That's the label cells were only successfully tracked ex vivo and immediately after injection into the rat brain parenchyma, which means the potential of the gold nanocomplexes for long-term in vivo tracking is still unknown, especially in multiple tissues of immunocompetent animal model. So the aim of our study is to explore whether the gold nanocomplexes can be used to label our PDL-SC, and whether this labeling technique can allow for long-term tracking after the cell transplantation into the rat at different tissues. To do that, we performed our study. First, the PDL cells were isolated from the extracted teeth. Briefly, the PDLs were collected from the middle third of the roots of the patient's extracted teeth and inoculated on a T25 culture flask immediately. Then the PDL cells were isolated from the teeth, and we cultured the cells for several passages and used for this study. Before we use the cells, we first characterize them as mesenchymal stem cells. It includes a series of experiments. It may sound complex, but you just know that they are flow cytometry, cell doubling time assay, colony forming unit, and osteogenic differentiation assay. These techniques are suggested by the criteria of the International Society for Cellular Therapy to identify the stem cell. And in the meanwhile, the gold nanocomplexes were synthesized to label the characterized PDL-SC. The cellular uptake and the labeling function of the gold nanocomplexes were simultaneously verified by bright field, fluorescence, and transmission electron microscope. After this, we know the cells can be labeled by the gold nanocomplexes, but before we use them, we have to check, are there any side effects of the labeling on the cells? And that's why a series of more experiments were performed to evaluate the side effects of the gold nanocomplexes on host cells. They are Alama Blue, cell viability assay, osteogenic functionality assay, PCR, and immunofluorescence staining. After all this investigation into the labeling function and side effects, the cell tracking were tested ex vivo to verify if the label cells were indeed visible enough by CT at different concentration or time. And then, in the final step, we finally can use the labeled cells in vivo to see if the transplantation can be visible by CT inside the living animals. Okay, let's jump into the results. First, the isolated cells were characterized as PDL-SCs, as proved by the following experiment. The flow cytometry analysis showed their specific surface antigen expression as stem cells. The colony forming unit assay and the cell doubling time assay showed their self-renewal ability. And alkaline phosphatase and alizarin red S staining showed their osteogenic capacity. So this experiment proved the cells we isolated from the extracted teeth were PDL-SC. And then, secondly, research showed how the gold nanocomplexes were prepared. The gold nanocomplexes were mixed with polyiolysine to neutralize the negative surface charge, so enhance the cellular uptake. Then the mixture were further synthesized with rhodomin B to render the nanocomplexes fluorescent. In this way, if our cells really uptake the gold nanocomplexes, the cells will emit red fluorescence under the excitation lights. It may help us to check if the cells already be labeled. And then the cellular uptake of the gold nanocomplexes were verified by TEM in a way of endocytosis. The fluorescence microscope confirmed the existence of intracellular gold nanocomplexes, and the fluorescence intensity were endurable for several days. Which means the gold nanocomplexes have a potential to stay in the cells for several days. And because the fluorescence intensity still decreased slightly over time, we wanted to know if it was because of the fluorescence quenching or because the gold nanocomplexes indeed influence the cell viability, which means the labeling agents may kill some of the cells. And that's why we conducted the last Alama Blue viability assay. And the results showed the gold nanocomplexes did not influence the cell viability for at least 7 days, which means it's quite safe to use the gold nanocomplexes to label our stem cells. And then, from the above experiment I have just mentioned, we know the gold nanocomplex did not influence the cell viability. But what about osteogenic differentiation capacity? Because we know the stem cells has a lot of capacity, like they can form the bone. But if we use the labeling agents, it might reduce the such capacity. That's not good. So we tested it in 3 different levels. First, the ALP activity assay and ARS staining showed there was no significant in osteogenic functionality between the labeled and unlabeled cells within 21 days, which means the gonadal complexes won't influence the osteogenic capacity. And also we do PCR and we do immunohistochemistry. Both results show the same conclusion that the gonadal complexes won't influence the osteogenic capacity of the stem cells, which is very good. And then we tested the labeled cells ex vivo by CT. The results showed the labeled cells could indeed detectable with micro-CT. And the CT attenuation of the cells showed an excellent linear dependence on the number of the labeled cells over a wide range. That means the labeled cells could not only be detected, but also be quantified with micro-CT. The CT attenuation could maintain at a certain level even after labeling for several days, just like the fluorescent intensity. So from now, the ex vivo scanning suggested a promising potential of the gold nanocomplexes for cellular imaging. Let's continue to see if they also worked well in vivo. To test the in vivo cell tracking function, the labeled cells were injected in rats by different routines to test their long-term cellular tracking. The rats were scanned in vivo at different time points to check the existence of the distribution of those transplanted PDL-SC. The cells were transplanted in limb muscle, submandibular angle, buccal mucosa, and the gingiva around the upper first molar. After 5 days, the Hounsfield unit decreased. After 5 days, they were still clearly detectable by micro-CT as hyperdense contrast areas in all injection sites. Although the Hounsfield units decreased clearly, it remains very easy to distinguish the hyperdense contrast areas from the surrounding tissue, even at 5 days after the transplantation. As you can see, the yellow dots around the tissue. The result is really good, but we still didn't know whether the hyperdense contrast areas were coming from the transplanted living cells or just the golden complexes coming out of the dead transplanted cells, or they are simply uptaken by the rat macrophages. So the final experiment of this study was immunohistochemical assay to testify whether the hyperdense contrast areas in micro-CT were exactly the living transplanted cells. To do that, we use immunohistochemistry assay. We used 2 markers, which is Ki67 and lamin AC, because these 2 markers are only expressed in human stem cells. So if the tissue we collected can express those 2 markers, it means the tissue contains our transplanted human cells. Of course, the result showed the tissue has these 2 markers, which means the hyperdense contrast areas from micro-CT were indeed coming from the living transplanted cells. Okay, conclusion. This study was the first to label PDL-SC, which rendered the cells detectable by micro-CT. It showed that golden complexes had no impact on PDL-SC viability and osteogenic capacity. It also allowed us to track cells in vivo for at least 5 days in immunocompetent rats. So this novel study allows for longitudinal follow-up with the same experiment animals at different time points, which proved strong internal comparison and avoid euthanasia just for ex vivo tracking such as histochemical assay, and thus can reduce the sample size. It may become a widely used approach for in vivo tracking with utilization of golden complexes for oral maxillofacial and systemic application of stem cell therapy. We believe this topic to be relevant and of interest for the oral health science community and also the clinics. Okay, thank you very much.